Greetings of the day to everyone. My name is Rishabh Kabra, along with my associates Upasana Anant and Ritish Kamboj, from 21 Integrated MSc Biotechnology at V.I.T. Bhopal. On behalf of my team, we would like to welcome you all, and it's our honor to deliver the explanation in front of you on a very interesting and fascinating Plasma composes 55% of blood, and rest 45% composition is of blood cells, which includes white and red blood cells. Blood contains cells, proteins, and sugars. If a test tube of blood is left to stand for half an hour, the blood separates into 3 layers as the denser components sink to the bottom of the tube and fluid remains at the top. The straw-colored fluid that forms the top layer is called plasma and forms about 60% of blood. The middle white layer is composed of white blood cells, WBCs, and platelets, and the bottom red layer is the red blood cells, RBCs. These bottom 2 layers of cells form about 40% of the blood. Let's continue further with Upasana.
Having read about the basics of blood, now let's hop to the main topic: erythrocyte sedimentation rate. Do you know the meaning of sedimentation? It's quite simple. It is the process where particles in a suspension separate out from the liquid and settle down due to gravity. Blood being suspension itself shows this property too, that is sedimentation occurs in blood when an anticoagulant like heparin is added, then erythrocytes settle towards the bottom of the test tube. The fact is, the fastest settling particles are larger, heavier, spherical molecules because the sphericity of sedimenting particles is an important condition for good sedimentation. And the slowest settling particles, which sometimes cannot be settled accurately or properly, are tinier, lighter, and/or irregularly shaped molecules. How do we calculate E.S.R? It is quite simple, and it varies with gender. Like for males it is age divided by 2, whereas for females it is age plus 10 divided by 2. How E.S.R functions. E.S.R basically a blood test that is carried to know the well-being of the blood system. An E.S.R can detect any disturbances in body like inflammation, etc. Why blood is chosen? As already told, blood is a suspension which is heterogeneous in nature. Erythrocytes are the main insoluble solute present, and it is these cells that perform the sedimentation, and the time duration they take to settle is called E.S.R rate. Important points. Different types of particle react different in this test. The action varies with the shape, size, and weight of the particles. For example, larger, heavier, and spherical molecules tend to settle faster and lighter. Tinier and irregularly shaped particles tend to settle very slowly or do not settle at all. Sphericity is an important condition which help in good and faster sedimentation. We went through the fundamentals of E.S.R, and now let's see how E.S.R functions with the help of an experiment. These are the things needed for the experiment: vegetable, tomato, petroleum jelly, vegetable oil, mortar and pestle, a glass container, distilled water, and a knife. The procedure is quite easy to understand. We will wash and cut the vegetables, then we will be mashing it by adding a little water. Then we will mix the mashed tomato with other ingredients, that is vegetable oil, petroleum jelly, in a bowl. Now let's relate this simulated protocols with the real company components. The fibrous tomato acts as erythrocytes, the vegetable oil can be related to plasma, petroleum jelly mimics the role of fibrinogen. We can also add other ingredients like butter that acts like globulin of blood, starch solution acting as WBCs. We can also create diseased conditions artificially. Example, using beet extract a reduced protein condition is reflected, and using beet shavings sickle cell condition will be formed. In order to perform the experiment successfully, it is very important to use clean containers, mortar, pestle, etc. Then we will transfer the contents into a glass container and wait for 60 minutes. Let's watch a video for a better understanding. Over to the calculation we get. After sedimentation, the measurement where tomato smash settle down is 2 centimeters. Time taken to settle down is 60 minutes. Rate of sedimentation equals distance traveled by tomato smash by time taken is 5 into 10 to the power of -6. For results we get: the vegetable gets settled down to the bottom. The oil settles above the oil. And petroleum jelly stays on top. There are 3 levels. In the first 10 minutes, RBC stalk over each other, called aggregation, a slow step. In the next 40 minutes. In the next 40 minutes, the aggregated erythrocytes settle down rapidly. In the E.S.R test, fluid is plasma factor. The particle factor is an erythrocyte factor. In the third and the final stage, lasting about 10 minutes, packing occurs is a very slow step. Now, Ritish will explain further.
So now we will understand why E.S.R is crucial. Because the numerical